Mid-morning. We're ready to go. We had a couple of gaps because of President's Day and so on in research grand rounds, but we're back with a vengeance and have a full program for you today. The menu today is shown here. So I want to start by talking about some new resources that we have and introducing you to that new resource for to help you with your the administrative aspects of your research. For the OIT questions today, instead of questions, I thought we might just talk about strategy, and I've got a diagram to show you that I think you'll find interesting. The main event today is Dr. Allen and Bain, who will present the latest in the discussions of a collaboration between a basic scientist and a clinician, and they will talk about their research on bone mechanobiology and tissue regeneration. Again, we think that the purpose of this is so that you can really understand what research opportunities are available, to see how other clinicians manage to work the interaction with basic scientists, and to see how hopefully the sum of 1 and 1 is 3. Also, we have the usual research process section, and today Leslie Meyer, who works for Drs. Manna, Chansky and Leopold at UWMC, has got a nice presentation to introduce you to the complexities of doing animal research at UWMC if you were to be involved in that. So let me start by introducing Teresa. She is right here. This is Teresa Bergholz. So Teresa is a 50% research coordinator, compliance coordinator, who joined us last month. And one of her tasks is to help you with smoothing the administrative aspects of your research process. We have been involved, many of us, with trying to overcome some of the hurdles to doing research. As far as you're concerned, all you really want to do is have an idea, design an experiment, and get on with it. But we all know it isn't quite that simple. And so part of Teresa's job will be to help you with the administrative aspects of doing your research. She has an office on the 10th floor, and she came to us from prior work at the University of Wisconsin and UC Irvine. Teresa, you want to just say a couple of words about what you did in the past and your research involvement?
Mainly I've been a clinical research coordinator at those two institutions, but I've also worked in the editorial office of the Journal of Applied Physiology. In my course of clinical research work, I've done a lot of work with the IRBs, so I'm ready to help you out with that, and learning a little bit more about grant submissions. So pretty soon I'll be able to support you in that as well.
So she's here to help you. She's a resource for you. You find her on the 10th floor. Stop by and say hello and share your problems, that is, your professional problems, with her. Not that you have any others. Her role, of course, will be coordination with the IRB, interaction with the Human Subjects Department. She's looking at funding opportunities, and she's going to help you with your resident grant applications. Okay, now you all are aware of the milestones we've talked about, particularly for the R2s, to move you through the research process. And I want to remind you that within the next month, the deadline is coming for you to identify a research mentor. We talked about this in the fall, about you looking at the landscape of research in the department, identifying a mentor, and hopefully a project. So I'll be interacting with each one of the R2s in the next month to ask you, well, what have you decided? And I hope there'll be an opportunity for you to briefly present your research idea to your peers at one of these meetings in the near future. So please get on the mark. Send me an email as soon as you know, and we'll be able to know who you're intending to match with. Now, it could be 2 people, it could be more, it could be that you haven't decided between A and B yet, but I really want to force the decision so that you're moving along and deciding what is it that you're going to do for your research component. The application deadline is the day after tax day for this year's research competition, and of course the equation we're interested in, resident plus dollars equals quality research. And so we hope that we can facilitate what you do by giving you up to $3,000 to do your research. I might mention to those lucky few who are the already recipients that you don't seem to be spending your money with great abandonment. So spend a little money soon, to those of you who've got research grants, because you know how money has the habit of going away if you don't use it, or somebody looking at it and saying, see, they didn't need it after all. So let's get moving and spend your money. If you don't know what to spend it on, I have a lot of ideas, so stop and see me. Now, we usually at this point go through a few OIT questions. But I thought that it would be interesting this morning to talk a little bit about your study strategy for the basic science component of the exam. And I've got an interesting bar chart here, which is actually from the OrthoBullets website. And it shows you the frequency with which each topic has been tested on OITE in the last 8 years. And there's some surprises here, I think, particularly about what is low on the agenda, and particularly fracture healing was interesting to me that it's so low. But let's look at it from the top. So orthopedic implants represent about 12% of the questions, or 25 times in the last 8 years, followed by osteopenia and osteoporosis, and surprisingly by rehab and prosthetics. And it seems to me that this perhaps doesn't exactly, and fourthly, of course, stat definitions. I don't know whether you're surprised by this or whether you knew all this already, but it seems to me that it gives you some guidance in your study preparation as to where you should put twice the emphasis than other topics. What is your impression about this frequency? Is this something you are well aware of? Are there surprises there for you? How could this guide your study? I thought we might have a brief discussion about that. Anybody got any thoughts on this?
Seems about right, but I know that the rehab questions were more common in the past. I feel like there's less of them now.
Mm-hmm.
Last test there was a ton of them.
Yeah, foot braces and stuff.
Rock bottom shoes.
Absolutely.
All right, I'm up for that.
Just glaze over those, I guess.
A.
It's always C.
So do you get, I mean, the ones in particular that it seems to me might not be mainstream for you are statistics definition and rehab and prosthetics. Is that accurate? It seems to me, where do you get that? Where do you get that information other than from your own reading?
Review book, you just memorize.
Books and
Yeah.
They ask pretty much the same things.
Same things.
Over.
alpha error, beta error.
Yeah.
Yeah.
Same questions.
Yeah, type 2 errors, type 1 errors, and so on.
Which is the sensitivity error.
Yes. And of course, all those definitions of positive predictive value, negative predictive value, they're things that's got to be second nature to you when you go into the exam. Of course, they're easy to forget afterwards, but they're things you really do need to study on. And I wonder if it would be helpful for us to have a couple of sessions on the areas that, I mean, I'm sure you get a lot of bisphosphonates, a lot of implant work, which naturally are the absolute top. But would it be helpful to have a session sometime in the future on rehab, to have one of the rehab people come and talk at this session? What would be most helpful to you? It wouldn't. You'd rather just know the questions and study the questions.
Yeah,
So
because honestly,
basic,
like,
it's-
there's 250
be
questions
offended.
on this thing, and in the last 8 years, they've only asked about that 18 times.
Right.
It's,
So it's not a
So-
big item for you,
yeah. I mean, it's, yeah,
It's like 1 to 2 questions per year. So yeah.
Yeah.
which you could afford to miss. Is that what you're saying?
It's also just, I mean, it's no, but it's like the questions are not like a rehab doctor. It's so beneath it. It's not even really. They ask us the same questions about like what implant would you put in this person's shoe, and I think no matter how much.
That'll be insert, not implant, but that's okay. No surgery on the shoes, please.
I would like to have somebody explain that stuff to me, because a lot of them, when you're reading it in the book, it doesn't make sense to me how that is biomechanically transforming forces in the foot, but
Yeah.
maybe that's just me.
I've talked to our
Yeah.
rehab docs about having them do a Monday morning conference, and there's nobody that's interested in
Nobody wants to do it. Okay, so
Nobody who does just like, what about says,
Yeah,
I
prosthesis
sort of, yeah, we could have like
They know a lot.
Yeah.
They even bring bagels.
You know, it's easy to forget that we have really quality prosthetics here. I'm sure you know, but I had this fantastic visit 2 months ago. I went to the Center for the Intrepid in San Antonio. I don't know if you've heard about this center, but it's a $160 million facility that's been set up for returning vets who are amputees. It is the best place in the world if you have an amputation. In the same building, they have a lab, a prosthetic shop, fantastic orthopedics, great rehab. Very tough place to go to. You go into a room of people your age, as many of you as there are here, without any legs. Very high amputees. You know, they've all been IED victims and so on. So it's a really kind of emotionally tough place to visit. But on the plus side, it's incredible to see what's being done for them. It's really remarkable.
Yeah, like maybe a basic prosthetics course,
Yeah.
like from foot implant to
Okay, let's try and do that.
just like
There is nobody in rehab that agreed to do it with us. Do you have the name?
I will dig for it.
You know, we can talk to prosthetics.
I think a prosthetist would be the best kind of person.
They actually know us.
Yeah, it's a combination.
buy stuff from these people. They
from
You can do a Monday morning
Yeah.
conference. It was a combo like Dr. Smith plus lab with the prosthetist where they actually show you how the stuff, you know.
We're gonna have one with Smith. We've got one for Smith set up, so.
Do you?
Bring a prostitute. You can try to bring in a prostitute. He'll bring us a
Yeah. And perhaps we'll couple that with just a trolling for all the questions that have been in the last 8 years and let you have those so that by the time you go into that session with Dr. Smith and whoever he brings, you'll at least be armed with those so that if you've got things you would really like explaining, you can. What about the statistics? Do you get enough on the statistics side, and who do you get it from? Google, yeah.
Like a little review is like, if like if we could just take the PowerPoint like the week before and kind of just fly through it and review it would probably be helpful.
Okay. So the suggestion is we get a PowerPoint with the main topics in that you have accessible to you for study purposes.
Yeah.
Or we could do it in this session too.
That'd be good.
Okay, that's good. Thanks. That's the kind of feedback I was looking for. All right, the main event. I think you're all aware of the Orthopedic Science Lab here in this building, and you are all aware of some of the work that Dr. Allen does with digit regeneration, but today you're going to hear what they have in common and what they're doing in this regard. Steve, it's all yours.
Is it on here somewhere?
It's right here, sorry, yes.
Sherry, I'm sorry I misspelled your name. Okay, you know, this morning, Chris and I just want to share some of the work that we've been doing that essentially is a sort of a fledgling collaboration between us and in the relationships or potential relationships between mechanobiology and tissue regeneration. And we're going to do that simply by providing an overview of a project where we didn't actually collaborate, but we shared a student. He had a student with money. We had a lab with a project. And so we put that together to look at a scientific question that was related to pathology, but which in turn is also going to be related to his work in tissue regeneration. So we'll try to show how those two things are connected. So I think most of you here know that in the Orthopedic Sciences Lab, our interests lie in this relationship between the role of physical stimuli, usually as are imparted by gait and locomotion, and how they relate to the regulation of bone homeostasis. From both a gain of function perspective, where bone adaptation leads to skeletal hypertrophy, as well as loss of function situations where you take away mechanobiology and folks lose bone. And so independent of whether we're talking about an anabolic or a catabolic situation, all of us know that osteoblasts and osteoclasts are involved in this interplay in terms of regulating bone mass. Over the last several years, what we have become very interested in is how muscle, the principal tissue in the body that is dealing with locomotion, is regulating bone mass. Is it indirect via its applied loads during locomotion, or are there other forms of communication between muscle and bone? And this is it's an incredibly important question for us to understand this because we know that exercise has great benefits for the skeleton, but we have we've as yet been unable to identify those things in the mechanical environment that are actually modulating these anabolic and catabolic effects. But we have tissues, muscle and bone, not to mention fat and nerve. These are all mesodermal tissues that embryologically and developmentally arise from the same stem cells. So at least when we're framing in our lab, that's kind of a common starting point for us, because it seems to be intuitive that during these developmental processes, these tissues would have learned to communicate with one another. So the model that we have used in our laboratory from a loss of function perspective was inspired by Nip and Tuck, that very popular reality show, where we just, some of the students in the lab just came up with this idea that, why can't we use Botox to inhibit muscle function and create a model of disuse? That was kind of the first sort of question that was asked by the lab, and it's turned into be much more complicated than that. But the bottom line is we can use botulinum toxin and its properties of preventing the release of neurotransmitters into a synaptic cleft to paralyze muscle. We use low doses, so it's a transient paralysis. But nonetheless, when you do this in our model, and in particular, we're using mice, over a period of weeks, we can study various outcome measures. We have the ability to study gait. So we have high-speed cameras, so we can look at kinematic and kinetic changes in gait after Botox injections. We can look at muscle volume. And the primary endpoint for us is what's happening in the skeleton. And so at various time points, we can look at these changes, and you can see, as an animal that's had their calf muscle injected with saline, and you compare that to a mouse whose calf muscle has been paralyzed with Botox, and you can see very profound changes, not just in the cancellous bone, but in the cortical bone as well. So several years ago, in fact, I was still in industry, I had some discussions with Ted and also Sean Nork. We were interested in, okay, so we know what's happening with Botox in this particular situation. We lose bone. But obviously in trauma, it's not, you have coincident injuries in muscles and nerves, which can have an impact on skeletal tissue. And so based on that, we came up with an animal model where we superimposed the transient muscle paralysis on top of a skeletal injury. And basically, what's involved here is we would inject the calf muscle in an animal who had a bone defect less than 1 millimeter in diameter in the tibial diaphysis of this mouse. And so when we first, there's a whole lot of things going on here, but the basic observation that was really surprising to us is that when we blocked muscle function, we eliminated the osteogenic response in the periosteum in these animals. So that implied several things to us, not the least of which is that mechanobiology, the absence of this muscle function, seemed to be having a very profound effect on tissue regeneration. The other aspect of this, when we saw this, we became very interested. We sort of made a serendipitous leap, so to speak, to this idea, well, I wonder if Botox would inhibit soft tissue osteogenesis as might occur in heterotopic ossification. So this is where we started to develop this link between heterotopic ossification as sort of an aberrant pathology with tissue regeneration, which is being inhibited by the transient paralysis of the calf muscle. So this is the normal response that you see, and then this profound inhibition. And so Chris, at this time, had a student in bioengineering, and they had a grant to look at tissue regeneration, a tissue regeneration opportunity. And so we put together this project to develop an animal model of heterotopic ossification using this idea of, how are muscle and bone communicating relative to the development of heterotopic ossification. And the reason that this is, I think, important is if you just look in the literature, as I'm sure many of you have, we know that patients that have spinal cord injuries, or, Peter mentioned this San Antonio Rehabilitation Center, where these veterans are coming back with IED injuries, head trauma, and massive trauma to their appendages. There's clearly a relationship between these neuromuscular trauma and the development of heterotopic ossification. And then more recently, there's some really interesting basic science demonstrating that disruption of BMP signaling in neuromuscular junctions is a causal factor for the development of heterotopic bone. And in 2007, a review was published, and this review was very interesting to us because DePaz and his co-workers proposed this idea that if you disrupt proprioception, if you disrupt neuromuscular signaling, that's what is leading to this anarchic, they called it, tissue relationships that lead to the development of heterotopic bone. So we circled back to our Botox observations where, Botox blocks not just proprioception, but it also blocks the motor neuron function. And so could we use Botox to block the formation of heterotopic bone? And so the animal model that we developed, or rather that Chris's student developed, was to take a bone morphogenetic protein, which there will obviously be questions on your exams about bone morphogenetic proteins, but we take a bone morphogenetic protein and we put it into a carrier material, and we can inject this into the calf muscle. And then over time, very rapidly, we develop a ossicle in the muscle that is going to mineralize and form bone. And if you know about heterotopic bone, it has all the same features as normal bone tissue. It's vascularized. In these particular models, it'll actually develop a cortical shell, and it'll have sort of a nascent bone marrow inside of that tissue as well. So this is it's a very standardized model for studying, for looking at heterotopic ossification. And of course, we're interested in this again because of potential relationships between neuronal signaling, muscle function, and bone formation, which are the same sorts of questions that Chris is asking in his research on tissue regeneration. And obviously, I wouldn't be talking about this if the experiment hadn't worked. So it turns out that transient muscle paralysis with Botox in this model attenuates the development of heterotopic ossification. So here we have an animal that has the BMP matrigel injected into the calf muscle, and in another group of animals we have coincident injection of botulinum toxin. So the muscle has been transient paralyzed, and we have a very significant impact on the development of heterotopic ossification. Now, in our lab, you know, when I talk about our research, I like to think of it as sort of a top-down approach. And what I mean by that is we have some really good in vivo models that we can use to understand sort of general applied questions of importance. But the next thing for this model, now that we've made this observation, is now to work upstream from this observation and begin to look at the cells and the signaling pathways that are involved in the formation of heterotopic bone. And it is these cells and signaling pathways that we're interested in that connect what we're doing with Chris's work in tissue regeneration. So, Dr. Allen, I think the next slide is you. We can either take questions now or we can wait till the end. We'll take a question now. Go ahead.
Have you taken that any further to see if there's any way to counteract the botulism? Like, is this an active muscle thing, a passive muscle thing? Is that to be potentially helpful?
No, we haven't. It's fair to say that we know that Botox is not having any effect on the bone cells, because I could take you back to that earlier slide where we show this nice osteogenic response in a normal animal. Botox inhibits that. If you inject Botox directly into that defect, there's no effect on bone formation. So we're pretty certain that it's a muscle-bone interaction. We don't think it's passive, simply because, you know, when you have a paralysis in an individual that's associated with soft tissue trauma, you develop heterotopic ossification anyway. So it seems to us that there's something in the muscle coincident with inflammation or injury that's being released. We like this model of disrupted BMP signaling at the neuromuscular junction. We think that that's going to be important, and so we just are going to drill down on that. We have plans to submit funding to the Department of Defense to try to study the cell biology, and hopefully with Chris's help we'll be able to get some of that money. Okay. Yes.
Does botulism affect regional blood flow? And paralysis of the muscle? Is that
well, I would think that blood pressure and things are probably going to go down in those regions, but we don't have any tissue necrosis or anything like that. And within 4 to 5 weeks, at least at the doses that we're using, the muscle function begins to rebound, which is another phenomenon that we're interested in, and we've identified some interesting signaling pathways that are involved in that. And for sure, you know, the fact that we get all this great heterotopic bone, which is heavily vascularized in a paralyzed muscle, means that we're not compromising the blood vessel formation to any great degree.
Okay, so I think Mike has cool stuff. Oh, another question?
Oh, you need the mic. No, really.
Trust me. You get the mic.
Oh, that worked well. Just hold on to it.
Yeah, that'll last for a minute. So that is cool. We'll get back to that in a minute. But first, having been an R2, 3, 4, 5 myself, only 1 year at each stage. Thank you very much. Let's pretend that Ted and Peter and Steve are out of the room, and don't camera the residents. So who's really certain they're going to actually do research in their career? It's very unlikely that most of you guys are going to go on and become full-on academicians, despite the fact that you really are at the top in terms of resident quality and background, which you, I am aware, think yourselves to be is actually the case as well. So granting that like 1 or 2 or 3 of you out of how many are here are going to do that, you must wonder why you have to do this. But can anybody tell me? I'm trying to not just feed everybody all the time. I'm trying to get more Socratic because I understand down the line it's going to help you to have been asked and have had to come up with an answer. Why the hell do this? So I've got a couple of reasons. I don't know if there's a great reason, but why should you even bother with a research project? Does anybody have any inkling as to why we would want for you to do that? Putting aside the very cynical but probably at least partly true reason is that we get free labor out of you guys, then we get to, you know, become more successful in publication. Can anybody tell me why it might be of value?
I think a large part of it is so that you have a better understanding of the research that is coming down the line. And
Yeah.
be directed
So
to
that's
the
one of my excuses as I try and torture you guys. So you're going to read all this stuff, and some of it's crap. And until you've done both good and bad research yourself, it's difficult to differentiate between the two. But if you spend any time in the lab, you'll do both of those. And reading the methods of papers that come out can help you understand whether what they conclude has any basis. And a lot of the stuff that's out there has no basis. You can't really trust it. And so it's important to know that, because whether you do research or not, you're going to go forward and do things differently by the time you are 10 years into your practice than you're seeing them demonstrated for you now. And a lot of that, we hope, is based on what we believe to be science, but not all science is good science. So doing science, whether it turns out to be good or bad, will help you understand the process. There may be some requirement from the academy that you do research. I don't actually even know if that's true. But I want you to look at this whole business as an opportunity to understand what others are telling you and to differentiate between good and bad. I know Seth does a lot of training in that regard. For the large number of you who said that yes, you would love to do academic research, if you actually find yourself in an academic institution, it's really a lot easier than if you're out in private practice, because there are a lot of smart guys running around, smart gals running around, who do this full time, the PhDs who you know here, and others who aren't in orthopedics, but who have tremendously active intellects and are trying to answer problems, but they lack what you have, which is the everyday experience with patients who have problems, and sometimes they can't connect the dots and understand what patients really need. So they can spend months or years trying to answer a problem that really isn't one, and it's helpful then for them to have a clinician who can tell them, well, you know, what we really need is this, actually, rather than what you think you're trying to do for us. So PhDs and in general are usually happy to have a clinician help direct their efforts. Somebody made what I thought was the funny quote that 2 years in the lab can save you an afternoon in the library. And I think that the same is true of an afternoon with a clinician, so that they don't waste their time answering a question that has no clinical relevance. And the last thing I'll say is that the advantage to you as a clinician who's also interested in research of collaborating with the researcher is that there are times when you cannot physically be in the lab, and yet the lab goes forward, and if you're doing anything basic sciencey at all, I screwed up a whole month's worth of my own poorly funded research by racing down from Harborview to the U to change the incubator O2 tanks when I was growing these little digits, and rushing back for an on-call case on a Friday afternoon and forgetting to turn the incubator back on, and digits don't do well at room temperature. So that was a lesson to me of the advantage of having at least a tech. And so going forward, if you can ally yourself with somebody or a group of persons who are there all the time, then you'll have fewer of those unpleasant experiences. So back to Steve's stuff. Is there a clicker somewhere here? Cool. How do we advance this? I'm going to go to the fourth slide. So I work, speaking of collaborations, I like Steve. I'd love to collaborate with him. I haven't really done anything with him yet. And we did share a student, but I have collaborated with a guy whose slide this is, and his name is Ken Munioka, and he is at Tulane. And that came about interestingly in that I was doing all this business with trying to regrow digits, and one of the guys in Bio-E here had asked me then to chair a session at a national regeneration conference, and I told him I haven't actually regenerated anything. And he said, invite smart people. So I got to do that, and one of the guys that I invited was this guy who's done a ton of fascinating work with mouse digit regeneration. And then he saw what we had done with human fetal digits and invited our little group along. And that down the line through a circuitous route resulted in a couple of years of Army funding for about $600,000. So you never know who you're going to meet and how helpful it will be to establish a good relationship with them. But as an aside, again, if you ever have a chance to talk to anybody in basic science, be nice and be polite and say yes as many times as you can in amongst your clinical responsibilities, because you never know which of those partnerships is going to result in something that's really satisfying and helpful. Long story short, to tie in with what Steve talked about, if you think about HO here at Harborview, you think about it as the enemy, and you're trying to carve this out and restore elbow motion or hip motion. But if you look at it from the flip side, it's extra bone. And if you're trying to regrow parts, extra bone would be great. It's going to be the foundation on which you build extra muscle and tendon and nerve and fat and skin. So Ken Munioka's interesting observation is that the BMPs, which Steve has spoken of, do stimulate extra bone, and you can actually direct extra bone in such a way that you may elongate amputated digits. And he's actually got fascinating work showing that BMP9 appears to allow for joint formation in a way that is not yet understood. But the long and the short of it is that we're hoping that with Dr. Bain, we can look at ways to not just impair or impede the progress of heterotopic ossification, but turn that on its head and try and regenerate bone and then regenerate soft tissue structures through perhaps the same signaling cascades. And I guess for your guys, from your guys' perspective, it's not important that we do this, but it's important that you know that we have projects like this floating around, and that these do have, to a greater or lesser degree, some little bit of money associated with them, so that your goal as a resident trying to do research is to make sure that you have a doable project you can complete in the timeline. And you have to sort of be vigilant about that, because we're all very enthusiastic about doing these things, but we're here for a long time, hopefully, and you guys don't have a lot of time. So as you hunt for a mentor, in the back of your mind, be vigilant not to get led down the garden path. So if you come to me and say, I want to regrow digits, I'll say that's great, and then we'll look for something you can actually do in your 3 months, because I've been doing this for 13 years, and we don't have any new fingers yet. So you got to really narrow it down. And you also ought to make sure that, in addition to the wonderful support that Dr. Kavanaugh spoke of, that there is funding from some source, because everything costs a lot more than you would think. And the most expensive thing is other people's time. And so that's going to be tech time. And so make sure that you have the foundation to get done the work that you want to do. I actually don't think there's a lot of value in talking about the specifics of the science of this, as much as I think there is in talking with you guys about the mechanism by which you can get plugged in. And so I do have a little bit of funding, and I've talked with several of you through the months and years, and I'd be delighted to talk with any of you in greater detail about projects with me. But my tendency is to sit and wander, so I'm going to cut myself short here and say that if there's anything I could do to answer a question that any of you have, I'd do that either now in the setting or later on when we're all even more bouncy and more wide awake. So why don't we shut it down with that, and I'll answer any specific questions now or hear from any of you individually later. Okay? Oh, yes, ma'am. Yes, Dr. Daly.
I was wondering if you've had any experience getting residents into your lab, and what's the most effective time frame for that? Because we now don't have a research rotation anymore.
So I didn't know that. I thought we had a...
Kind of carving out time here and there. during, like you said, basic science is very rigorous work. I don't know, have you had success with resident partnerships? Has it usually been in that 6-week packet of time?
You guys are the smartest and hardest working people I've ever bumped into, but I've never had a great long-term opportunity to work with a resident because the folks who have come to me have defined chunks of time, a long time. I get a lot of folks from the I'm actually, for whatever reason, an adjunct associate professor in bioengineering. So I have access to their students, all of whom have an even more firm mandate to do research in that they have this capstone project. Every one of them has to complete basically a thesis with a project, and they come with money and time. And so they just camp out in the lab, and I co-mentor with somebody that's a full-on bio-E PhD. So that works well, and it's exactly useless to you to be able to try and follow that path. We'll have to work on that because there are...
Yeah, we need to regroup about how this is working out, the children's rotation, and if it's working out. This is really a very significant issue. So perhaps next time we could just have we'll take time to discuss that.
I'll just say one thing in closing, that one of the things that we have going on now has associated with it a collaborator at a different institution with some of those bio-E students. So there's no reason why you can't be a co-author on at least an academy submission with 3 or 4 or 5 or 6 people. You don't need to be the lead author, and you don't need to be doing all the work. But if you're involved, then you get the advantage that Paige spoke about. You know what a project looks like from its initial construction to hopefully its completion.
Chris, thanks so much. Very useful perspective. All right, the final event today. Steve.
Just the last thing. We have projects. Thank you. What's the related to heterotopic ossification that are ongoing right now. And Chris and I, in our own discussion, have come up with an idea to develop a regeneration model and which we can really have a mentor in the lab to work with a resident on that type of project.
That's good to know. So there's a potential project if you're looking for an R2. Yeah.
For the R3s, between now and next month, is there a way that you can keep track of what you are doing on your research day? And if it's not research, what it is you're actually doing, either babysitting the ER, getting pulled into someone else's OR, someone's clinic, and then we can actually look at a month.
Well, we can bring those numbers next month.
Yeah. If it's not working, then something has to change.
But actually, I will not be here next month. I'd like to do that in the May Grand Round. So if we could do that, anybody who's at Children's now for the next 2 months, would you just kind of log your research days? Just make a note.
So Peter, do I have it right that it is they are compelled, these folks are?
Yes, there's a requirement for the residency program to be engaged.
But no time allotted for it.
I think as time passes, the attendings over there are forget how important it is for us. That this is really our only time to have dedicated days to research. So we do end up covering, most of the time it's covering OR. OR days that may have been previously on schedule that are now scheduled, and, or being on call and being there in the morning for sign-out roundings before stuff, and then coming back.
Peter, I'm just going to advocate for these guys. You can never say no if you're a resident and you're invited.
Oh, I know.
And clinical opportunity.
You cannot. But you know, we can't afford mission creep on the research days where it becomes now less of a priority, suddenly not even a component. So I think this is certainly a warning bell for me that things do not seem to be working out the way they were intended. It sounded good at the start, but it sounds like it's not working out.
Because you guys do it on joints too, right? You're supposed to still have a day.
We did not.
Technically, no?
We did not. I think you know. Friday, but it turned out sometimes you were called here again, so if you go, you're around twice a day, you have to cut down to nothing.
If you want to share emails with me about this, I would be, and I'll keep them in confidence as to who they're from, but certainly I need to get this whole picture in front of Dr. Chapman and tell him that it's not working out.
And I think the VA, there's no reason why there can't be a research day, like on the Tuesday when there's one OR.
Right.
And 5 residents. There's no reason why that we can't.
There's no need to spread the load on a day like that when you could concentrate it and give you research time.
Right.
Yeah.
What, I mean, and if we just kind of went ahead and said,
It's just hard when, you do take that one day and nobody sees you around, and pretty soon it's like, the attending starts speaking of that day in jest, or, track where you were and start asking questions, and then pretty soon it's just like not worth the hassle to be out of clinical duties.
Well, you need to be. That's the only way, because as Chris says, if you're there, you cannot say no. So it needs to be that you're not there.
I mean, an 80% success rate is probably good enough for all of us to get research done, but as soon as you allow it to be that, then pretty soon it's, well, what about 70% and then 60? And then now this year, in a 12-month block, it will be less than half.
well, then the other problem is it changes every 5 weeks what day of the week it is, so it gets really hard to coordinate
It's hard
with your
to
mentor.
coordinate with your mentor, yeah.
Because it's Mondays 1 week, 1 month, and then it's Thursdays the next month, and then it's half a Wednesday and half a Friday the month after that.
Saturday and Sunday.
Sometimes.
When you're not on call,
Yeah, all right, let's, Leslie is up with a discussion of animal research.
Hi, my name is Leslie Meyer, and I'm the admin assistant for Doctors Chansky, Leopold, and Manor, and I've met a lot of you and seen the rest of you, and it's good to see you here today. We're going to talk a very little bit about very brief, basic idea of how animal research is conducted at the University of Washington, and I'm open to getting any corrections on mistakes I make, so please feel free if I've got something hideously wrong up here. There are basically 4 areas we're going to talk about: animal research management, the requirements for conducting animal research costs, and resident involvement in animal research. There are 3 groups that basically rule over animal research: the Institutional Animal Care and Use Committee, known as IACUC, which reviews all animal use protocols for research and teaching at the UW. The Office of Animal Welfare, the OAW, provides administrative support to the IACUC, coordinates these reviews and manages animal use training program. And the Department of Comparative Medicine, which manages the day-to-day operations for animal procurement, housing, and medical procedures. When you're going to create an animal use study, you fill out a form called the PRF, the Project Review Form, and that's found on the IACUC homepage. A PRF is submitted to the IACUC by a faculty member. Residents cannot conduct independent animal research, and residents can be listed as co-PIs on the PRF. And this is the site where you would find everything. It's a secured site, and due to time constraints, I'm not going to go to it and show you its tricks, but you log in using your UW Net ID and password. This is what the face page looks like, and you can see that it has links to the Department of Comparative Medicine and to the Animal Use Training Program. And it's a very comprehensive site where you'll find under contacts all the people who are listed in the different committees for your animal research, the admin contacts, the department contacts, where all the forms and the policies and procedures are, and other additional information that will be helpful in the process of doing animal research. This is what the forms page looks like. You would download a PDF of the project research form, or the PI, what I should say. Once the resident completes all the training that's required, they would be added onto the protocol with the request for addition to new personnel to animal use. Very awkward phrase there. But that's the form that the PI would fill out. And the requirements for doing animal research, I think, are as difficult or even more so than doing human research. There are a great many exams you have to take and complete. And as you can see, you must complete federally required training prior to inclusion on a PRF. And what is the training all about? There's an online exam called Animal Use Laws and Regulations. There's a form you fill out, 2-page form, the Animal Use Medical Screening Form, to make sure that you don't have any diseases that could be communicated to the species you're working with. Animal use classes and certifications, which is both online and hands-on, specific to the species and procedures you'll be working with or performing. And this is the Animal Use Training Program site. Again, due to time, we're not going to go there, but you can log in and look at it and see the variety of protocols and exams and research material that are available to you online. Okay. Animal research costs. The Department of Comparative Medicine at the UW manages animal care and housing for animals on campus. They charge a per diem for each animal to cover housing, food, and general care. All procedures for the research project are charged separately to your research budget. And this is a cutout from their website that has all the protocols and per diems listed. And so one cat costs $22.01 a day. Most studies will use more than one cat. And you know, for those of you who don't like cats, you could probably use a lot of cats. Goats? Goats are $10 a day. Well, they need a little less care. You don't have to like play with them as much, I guess. I don't know. Moving forward, that's just for the housing, feeding, and poop scooping for the animals. For buying the animals, that's another story. A common mouse runs between $15 and $25 each. If you're using a transgenic mouse or a mouse of any other very, special breed, it can run up to $1,000 per mouse. What? $1,000 per mouse. Special mouse, more special than ever. Yeah, and just ridiculously expensive. Rabbits, on the other hand, tend to be a little bit cheaper, and they're between $40 and $60 each, depending on whether you need a retired breeder, if you need a male, you need, babies or whatever you need. Pigs are sold by weight, and they can be between $150 and $250 each. Goats, same thing, between $200 and $300. Vendors will also charge a shipping fee, and the UW, bless their heart, will charge you a $20.97 fee for processing purchase forms. It's wonderful. This is an example of housing and surgical services costs for the initial phase of a proposed rabbit study using 54 animals, including the following. This study takes place over a period of about 2 years and assumes that the 54 animals will be in-house every day. It's kind of a high estimate, but that's what you do when you're looking for money. Animal costs, including acquisition, per diem, and surgical time, is about $25,000. That's a lot of money. Histology for each animal for this particular study runs out to $21,600, micro-CT at $200 per animal, $10,800, labor and observation at 187 hours. This is for 1 hour for 54 animals each at $10,087. And your post-procedure, or that's for post-procedure 1 hour watching the animal. And your total costs estimated for this study are about $68,173. That's for this one part of the study. This does not include the implants that they were putting together for this study, does not include the PI salary, it does not include a lot of other things. So animal research is not cheap. So Drs. Bain and Allen talked about how to get involved, and how you do that again is you identify a faculty member with an active animal research study you can join inside or outside our department. In the other option is identify a faculty member who's interested in designing an animal study and defining your role as a co-PI and working with you to research funding opportunities. One place you can go to look for opportunities is the Institute of Translational Health Sciences, and that also is a secured site, I believe, and you can go and check it out and see what research opportunities. And of course, your greatest source for research information in our department is Dr. Kavanaugh. And if you have any questions, I'll be glad to answer them. No? Thanks. Who wants a bunny?